So my course project was looking at mutations that are associated with COVID-19 prevalence. And I learned how to use the haplotype caller pipeline to process genomic data, and then additionally how to write different functions to annotate PCF files. And from this, I essentially learned how to go from a very large data set, be able to write different functions to process, annotate, and then come up with solutions.
Our course project looked at lactose intolerance and what genes were relevant to lactose intolerance. I think the biggest thing I took from the course project and the course itself was the experience, the hands-on experience that I had on the Google Cloud VMs, so that I could apply these in other classes and my research as well.
It was me and two other students who were really interested in long COVID, especially Dr. Michael Snyder was a guest lecturer twice, who had a really interesting project. He presented on wearable health data and how you can track simple biomarkers and predict things with more accuracy than current methods of like developing COVID and like the possible health effects of long COVID. And we thought it would be interesting to visualize doing a project for something that is not yet fully understood, and to see if we could gather current biomarker data from those wearables that Dr. Snyder presented on, to see if that indicated future symptoms of long COVID. So we went through that. We decided to implement it through Google Cloud, and we took a look at some of the possible functions of Google Cloud we could use. We ran a scalability and a cost analysis. So it was a really interesting way to see a project that will probably be done in some respect by someone, or is currently being looked at, and how what sort of money gets invested into those things, and what things have to be considered, again going back into the patient health information and protected data, and justifying the use of that in order to understand something currently pressing in like the medical community.
My course project is about a chronic skin disease called psoriasis, which affects millions of patients worldwide. And in this project, we're analyzing genomics data and also RNA-seq data that might provide more insights into this disease. I learned how to set up analysis pipelines to do the analysis, and also I learned how to test the scalability of a system, and also do the security analysis and also the cost analysis.
Our course project, I was working with a PhD student in genetics, so it was a lot more than I could have come up with on my own. And I think that was great for my education. It was single cell RNA comparisons, and essentially it's just how you could use just DNA from one person taken at different spots of the body to see like maybe some cells are like behaving funny in different areas. And you could use this for like diagnosis, or see the impact of like different environmental or biological things going on with the person.